Hello, my name is Caio, and I'm from the University of Campinas from Brazil, and I'm showing my project entitled Occurrence of Fusarium graminearum species complex and its mycotoxin barley naturally present in the pampa biome. So, the Fusarium species, especially Fusarium graminearum species complex, affects small cereal grains like barley and wheat. And the main concern about Fusarium graminearum species complex is because this fungus can produce mycotoxins known as deoxynivalenol and zearalenone. And these mycotoxins and the fungus can affect the cereals, the barley. And another problem is that we can find, and some previous studies reported that found Fusarium species in native cereals, in native plants, and these native plants, can serve as reservoirs of Fusarium species, of mycotoxigenic Fusarium species, and this fungus can affect the agriculture as well. So the objectives of this project is to characterize the Fusarium graminearum species complex isolate regarding the genotype and to evaluate the occurrence of DOM, triadone, 50adone, NIV, D3G, and ZEN in native barley samples from the pampa biome of Brazil. So the materials, we collected 48 samples of native barley from Brazil in the south region, and we isolated the Fusarium graminearum species complex. And to characterize this isolate, we did the phylogen using the molecular markers of RPB1 and RPB2, and we did the genotype profile of this isolate regarding if these isolates are 15adone or 3adone or NIV genotype. And for the mycotoxin analysis, we did the extraction by the Quetcher's method and injected in the liquid chromatograph coupled to mass spectrometry. So the results, we have the phylogenetic tree, and here it showed that the majority of the isolate we had is grouped to Fusarium graminearum here, and all these isolates of Fusarium graminearum strict sensu is 15adone genotype. And we had as well isolates that's grouped with Fusarium mesoamericanum, Fusarium cotederiae, Fusarium austramericano, and Fusarium asiaticum. As well, we found a new group that grouped to Fusarium nepalense. And all isolates of Fusarium mesoamericanum, cotederiae, and asiaticum was NIV genotype. The only isolate of Fusarium austramericano is 3adone that can produce 3adone. And the occurrence of mycotoxins, we found that the samples was highly contaminated with DOM 3-glucoside, and triadone. And we couldn't find 15adone in the samples. The hypothesis is that the isolates that produce 50adone convert the 50adone to DOM during the biosynthesis pathway, and That's why we found DOM. And 3A DOM, we found we had a high contamination of 3A DOM. Otherwise, we had only one isolate of 3A DOM. So I think the problem is during the isolation of the fungus. So we cannot find, we couldn't find an others isolate of Fusarium 3A DOM. So we saw the co-occurrence of these mycotoxins, and some samples were contaminated with D3G, DOM, 3A DOM, and NIVI at the same time. The hypothesis that we found high contamination of D3G is because of the plant. Maybe the plant converted the DOM and metabolized the DOM in D3G, that DOM complexed with the glucoside of the plant and formed D3G. So as the conclusions, the results have shown that native barley are potential reservoirs of this toxigenic Fusarium species and could possibly convert DOM in D3G. And further studies are needed to understand the interaction of the Fusarium graminearum species complex with native barley and with the ecological dynamics and of this important group of fungi, and to contribute to the control of this mycotoxin. So that's it. Thank you so much.